Good afternoon, everybody. I am Anisur Rahman, D1 student, Laboratory of Bioscience Engineering, Kyoto University. Today, I will be present my new research project. I am working with Shirataki-san. We are working together. The research title is Development of Measuring Technique for Freshness of Fish using Ultraviolet Image. Outline of presentation. The outline of presentation is background. I will be discussing some background, current approach, which approach now in use in industry, fisheries sector. Then our approach, what is MAAS, our hypothesis. We did some preliminary experiment. We will be showed material instrument use, method, result of the experiment, finally conclusion and next plan. Background. Fish is highly perishable commodity. When new fish is caught from sea or ocean or river, it is sweet flavor which is highly appreciated. When it is stored, the flavor is degrade and it is lost. And at the further storage, the flavor is all lost and it is deteriorated and it is not unfit to eat. So it is deteriorated. The freshness describes the presence or absence of deterioration. So it is needed to know as the amount of deterioration since they caught the fish caught. The traditionally freshness has been assessed by sensory method using the human sense of sight, smell, and touch. This is very quick method and undestructive method, but it is needed to train people. Current approach, which is used in fisheries sector. Firstly, sensory analysis, which is based on the human sense, the smell, touch. If it is very lot of fish, then it is difficult to measure the freshness of fish. Microbial methods use the growth of spoilage microorganism on fish based on the microbial method. Volatile compounds based on the fish gases. They use the gas sensor to determine the freshness of the fish. Lipid protein ATP use the K-value. Physical properties use the color and texture properties of fish. When it is long-term storage, the texture will be broken and this use the physical properties. Electrical properties, some electrical properties used for the freshness measurement in fisheries sector now. The disadvantage, the current approach has a many disadvantages. Some of the disadvantages, it is time consuming, expensive, and need to cut samples sometimes to measure the freshness of fish. So what we want fast, cheap, high accuracy, non-destructive method to evaluate the freshness of the fish. Our approach. Fish is exposed by UV in water. UV solar radiation causes directly or indirectly damage tissue in fish body. So fish develop the MMS materials which absorb the UV and minimize the structural and physiological effects on his fish body. The MMA is stored in the fish eye, lens, skin in order to protect from the ultraviolet light. What is MAS? MAS microscoping like amino acids, which is known the nature, sun, skin, water-soluble nitrous compound, which is characterized by cyclohexanone and cyclohexamenine ring conjugated with the nitrogen substituent of amino acid, who is protect from the photochemical effect of the solar radiation. MMA has the UV absorption maxima in the range of 310 to 362 nanometer, which is lay on the UVA above UVB. Another characteristic of a MAS can be degraded by bacteria very easily. Some example of MAA, this is the Eusozirenin 375 nanometer maximum peak and the Urohalothoris 362 nanometer, Serorine 334 nanometer peak, absorption peak. Total 19 MAS had been found now. Our hypothesis. MAS is decomposed by bacteria that makes up the MAS change in fish with passes of time. So, MMA has an absorption band of ultraviolet light with different peak respectively. In order to the change in time, there has a high possibility to shift the absorption peak wavelength. With this, using this phenomena, we want to develop the measuring techniques of freshness of fish. We did some preliminary experiment. We used the fish which is caught from the Biako Lake, Otsu. The scientific name of fish, Lepimuris Marco Sirius, common name is bluegill. We use for spectra data acquisition, we use this spectrophotometer, which is made by the Shimazdo company. Methods. The following procedure used for our experiment. At first we dissect the fish into cornea and lens. Then we prepare the cell for spectroscopy. This is the cell for lens, this is for cornea, this is for reference cell. We use the cellulose tape to attach the cornea and lens with the spectroscopy cell. We measure the spectra every 1 hour intervals up to 24 hours. After 24 hours, take spectra per day up to 7 days. After every measurement, we keep this cell in an airtight polythene bag and keep in 20 degrees Celsius water. Absorption of raw cornea. I think it is not readable. But we see we can minimize this the 0 hour, 12 hour, 24 hour, 48 hour, 72 hours, 96 hour. We see that there have a small absorption. The 358.6, 358.5 nanometer, there have a absorption. After some times, there is the after there have a this is disappeared. But why, I don't know. I need to read more about this. This is the transmission spectra of cornea. Here the transmission peak, higher peak, we got the 0 hour, this is the 358 nanometer, and after 12 hour, it is shifted to 362 nanometer. So there have a possibility to, there is a 0 hour, there have a peak transmittance 358.5 nanometer. After 12 hour, it is shifted to 362 nanometer. This is absorption of lens. We minimize the time, the 0 hour, 12 hour, 42 hours. We have a absorption in the indicate area, but I don't know why the figures like that. Maybe some error. I need to do more experiment. This is the transmittance of lens in raw data. This is the transmittance high. After that, it is reverse. Why? I don't know. This is the transmittance of lens. Here we see this is 0 hour. There have some noise, and this is the after 12 hours. There have some absorption in there have some belly in transmittance. There is a peak. So conclusion: we have a possibility to find the MMS at the wavelength 358 to 362 nanometer. Further to know details of the MMA, we needed to the SPLC experiment for the fish cornea lens. And before SPLC experiment, we will be do repeat experiment to minimize this error and analyze the spectra data. Thank you for kind attention. Give me suggestion to minimize the error.
I saw your spectra data. It is
yes, baseline.
You did.
I did some portion did, but not fully. I'll be present Thursday group meeting.
So you cannot saw this land effect removing today.
Yes.
Anyone else?
I just want to, if you want to, as I mentioned before, if you want to find the peak,
Yes.
the real peak of your,
Yes.
if MAAS is exists in that
Yes.
bluegill, so I'm not sure in that original data you will find something. That's why I think.
I did one, the absorption of cornea, they have a peak, and 70 days they have a peak. 362 nanometer, they have a peak. The error minimizes the baseline.
In second derivative?
Yes. I'll be sure to you.
So in the second derivative, you did the second derivative and then you fit
Yes.
it in original?
No, this is all raw data.
No, you saw the original. You saw the original, the peak of your original data. You saw us that peak. No.
This is the absorption of cornea. I use the absorption of cornea.
No, you mentioned some peak here.
This is the transmittance.
No. You mentioned some peak shift from this peak to
Ah,
this peak. Yes.
this transmittance of cornea. They have a peak, it is shifted after 12 hours.
Easy. This is
0 hour.
0 hour.
Oh, this is 24 hour.
24 hour.
Hour after 24 hour.
Then 12 hour is here.
Here. Maybe some things.
48 hour is which one?
48. Maybe, yeah. This one.
This which one?
Yes. Similar color.
No. You change the type of
Color.
No. Type of this
Line.
Like
Okay.
dot line or some sheep line, thin line.
Okay.
Based on the some difference in some papers, they say how those MAA change.
Yes.
How shift from 360 also to shorter wavelength.
Shorter wavelength.
That's absorbance or transmittance?
Absorbance.
Absorbance. So not this one.
Not this one.
So would you change it to absorbance?
This is the absorbance. Yes.
If MAS is included in this, maybe some absorbance here would be
Shift
changed
here.
this way.
Yes. This way.
Then which one is that? Oh, this one.
Yeah.
Zero hour.
0 hour.
24 hours. 0 hour. Transmit.
Absorbance.
Absorbance. But we don't know. This
Will contain the MAS. No. We don't know.
Something changed or
Something.
not.
When you prepare the samples, so every time
No.
before,
No.
no, I mean before your for example, the first from 0 hour.
On
then
sample.
It's only one sample.
On
but
sample.
I hear that before you measure the next measuring, you have to.
After every measurement, we keep the sample cell in airtight polythene bag and keep storage in water. After every measurement.
Every measure. So
Then when we will be measured, out of from water and measure this data, take this data and again put in water.
So I just worry the relation between that sample to the water is changed by time because of time pass away and then.
But water temperature is 20 degrees Celsius, not change.
No, I mean if your sample holds some water, so maybe because you.
But we use the reference as a air, but cornea or lens or gel, you may be some water. So we will be trying next time reference. We will be thinking.
It looks maybe short time. In short time, they may change.
Yes, within 6 hours.
Why don't you measure the every 2 hours, every 3 hours?
We measure the every 1 hour
every
data.
1 hour. Okay, which one?
This is every 1 hour.
Which one is it?
It is difficult to read. This is the 1 hour to 7 days.
No, maybe you can so one by one.
One by one,
and then
1 hour, 1 hour.
Here, one.
0 to 7 days.
Which one is the
This is maybe 0.
R is zero.
0.
0 and
This is
1 hour. Here, 1 hour.
or around here, yes. We need to.
Please
Okay.
show us another way.
So before you mentioned several kind of MAS. So that MAS we may find the pure of that MAS.
Pure MAS, I don't know.
For example,
From shop.
serotonin and then
We measure the
if-
spectra.
No, if you can find that pure, so your system should be detected or not that kind of spectrum.
Ah, yes.
I think if your system could detect this MA and the peak is okay based on your reference.
Okay. I will be fine.
If these are the same group, something changes.
Yes.
So maybe the baseline effect
Baseline.
because they did not remove the
Baseline.
baseline.
Oh.
Before
But,
I-
if he take some ratio, that the 280 and the 360 or some ratio might be changed. Anyway, that would be very curious and interesting. Anyone other suggestion? Okay, show us another way. Thank you very much.